Good afternoon and welcome to ATP-Bio's public webinar series. My name is Ellie Gardner, ATP-Bio's Communication Manager based at the University of Minnesota. Before we begin, today's webinar is being recorded and will be available on ATP-Bio's YouTube channel. It is now my pleasure to introduce today's moderator, Dr. Tim Pruitt. Dr. Pruitt, the floor is yours.
So thanks, Ellie. In today's webinar, ATP-Bio's Ethics and Public Policy component is very pleased to welcome Dr. Lakshya Gangwar to present One Field, Many Terms. Can cryobiology agree on its language? I'm Tim Pruitt, transplant surgeon at the University of Minnesota, and I serve as the co-director of the Ethics and Public Policy component. We work to promote the ethical development and deployment of ATP-Bio's advanced biopreservation technologies, and I will be the moderator of today's webinar. By way of introduction, Dr. Gangwar is a postdoctoral associate in the Department of Mechanical Engineering at the University of Minnesota. He's also a scholar trainee with ATP-Bio, an incoming fellow with ASME ECLIPSE, and a cryo member of the ASME Thermal Medicine Standards Committee. Dr. Gangwar is the lead author of a recent paper that was published in Cryobiology entitled The Need for Harmonized Terminology in Cryopreservation to Support Reproducibility, Regulation, and Translation. Lakshya, with his co-authors and senior authors, is part of ATP-Bio's multidisciplinary team of engineers, clinicians, ethicists, lawyers, and other experts examining the urgent need for harmonized cryopreservation terminology to improve reproducibility, regulatory clarity, and ethical governance in the translation across medicine, conservation, food, and agriculture. Today, Dr. Gangwar will present key findings from this paper for about 15 minutes, and then Dr. John Bischoff and Susan Wolf will each offer a brief response before we open the floor to question and answers. To ask questions, please use the raised hand function on the Zoom, and I will call on you. It's now my pleasure to welcome Dr. Gangwar.
Well, thank you all for joining. Good afternoon, good morning, wherever you're joining from. Myself, Lakshya Gangwar, I'm a postdoctoral associate in mechanical engineering. I'm also a scholar trainee at NSF Engineering Research Center, ATP-Bio. And I'm also working in ASME, ECLIPSE, as well as Thermal Medicine Standards Committee. So today, if you're here, you are in the ATP-Bio's webinar series, which is sponsored by Ethics and Public Policy pillar. And the whole center, as well as this webinar series, is funded by NSF. Today's talk would be on one field, many terms. Can we all cryobiologists agree on its language? It is an interesting one, and I'll give some slides and give our research about it. Then we'll open as a panel of Dr. Wolf and Dr. Bishop, which will give their thoughts, and you all can use that time to provide your thoughts as well. All right, so a little bit of why do we need to talk about this is because of the rapid advancements in cryobiology field which are happening. And to just give you a snapshot of it, for example, in organ preservation, all the big journal as well as news reports, such as New York Times, covering for partial freezing of kidneys, are talking about bringing back life from cryogenic freezing. Or we're talking about something such as using AI and for CPA discovery and really moving the field of cryobiology forward. Going on conservations, people are able to cryopreserve endangered species biomaterials such as eggs, embryos, as well as sperms. And then really thinking about, can we use cryopreservation as a tool to conserve and create biorepositories on even as far as moon. So all of these media is covering cryobiology in their own way, and it is really advancing. So that becomes that public is starting to know about that there is a field of cryobiology and cryopreservation technologies have societal impact in their lives. To just give you a snapshot of really past, this hasn't been happening now. Cryobiology has made advancement way back in time. So this is an article from New York Times about a frozen kidney restored a canine kidney. And interestingly, it happened at University of Minnesota as well, back in 1970s. So popular media in the country as well as outside US covers cryobiology topics once in a while, and what impression of cryopreservation technologies is sort of communicated to public is very important. And to give you an example of it is that was advancement happening, but to see these are also covered by scientific journals, whether it's supercooling of livers or vitrification of kidneys, or talking about vitrification of coral sperms. Nanowarming is one technology, and even going into field-specific journal covers. And these are not comprehensive, but to show you that cryobiology is not only making cover on these media pages, but also these scientific journals out there. So a lot of researchers from other fields also are having interest to come and learn about the field. But that brings into a question, is the language for the field is being used consistently, and can everyone learn about these rapid advancing technologies? So for example, this was a joke I was having with one of my colleagues. Is it sub-zero or sub-cryo? And to a person who's non-cryobiologist, and even to a cryobiologist, this might be confusing in that layman term of what does cryo actually means. So to start with this cryo adjective, let's break down the history of it. Cryo is derived from cryos, which is a Greek word, and it means icy cold. So the both things are important, icy and cold, because cold can be anything lower than a physiological temperature of body. 20 degrees Celsius can be called as colder to human body, but it is not icy cold. Ice is forming at a specific temperature, which is 0 Celsius, 32 degrees Fahrenheit. So icy cold becomes things around that temperature range or below it. Now, if you use the cryo adjective, you might hear in, is it cryonics? And that could be a science of freezing human bodies. Or is it cryogenics, which is the science of creating low temperature, cryogenic temperature, which is defined as less than 150 Kelvin by Cryogenic Society of America. Cryotherapy, and now you could have cryosurgery, cryoablation. Adjective remains the cryo, but those two are totally different than preservation. We are talking about destruction of tissues, and most of the times it is for preventing from tumor growth in cancer. And a really new thing, which is cryotherapy, but it is for rejuvenation and leisure purposes, such as the spa. So if you talk and you're saying, I'm going for a cryotherapy on the weekend, someone might be thinking you're going to an operating theater, but that's not the case. You're just going to the spa down the street. And then coming to cryopreservation, which is still the same adjective of cryo, but you're preserving things which are reversible preservation, where you have viable and functional aliveness in a tissue, cell, organ, whatever the preservation material is. Just to do an exercise of this, I typed cryogenic on Cambridge Dictionary, and you can see the two different or diverging definitions. One is about creating low temperature, which is what cryogenics is. The other is about freezing dead human bodies at low temperatures, which is what cryonics is. So a dictionary can mistake cryogenic for cryonics, then definitely others can too. And if you look at the field specific for cryogenic definition, you can see lower than 120 Kelvin, generation operation of those cryo temperature. So the general audience definition on the top versus the field specific. You can see the divergence in them. So what that leads to is a lot of confusion, and not only just in public, but it could be within the field specific experts such as cryobiologists. For example, vitrification, is it truly ice-free, slow freezing? Does that mean there's ice and glass, isochoric vitrification? Can there be ice in glass outside the sample, or partial freezing where there's ice in liquid? So a lot of these technologies are using these complex different state of matter, and that could be confusing. So I use AI, which is ChatGPT, for example, to create. This could be a new student or a new researcher coming to the cryobiology field who is really a heck-lot confused by just all the terms which we are using interminglably without thinking like what the implications could be. And that leads to, in the papers, one term becomes a trail of series of paper calling, for example, ice nucleation as ice crystallization. And then you end up thinking, is nucleation and crystallization the same thing, or is it something else? So a closer look at these definitions, just the very basic would be cryopreservation itself. If you look what could be the sources of confusion, that, for example, the cryopreservation could imply cold temperature means preservation, but in reality, control of these different factors is what giving the preservation the shelf life, meaning then those multiple factors could be CPA toxicity, osmotic shock, ice damage, fracture failure. Something else is, for example, sometimes definition might use cryopreservation as all biological materials, whereas other might use simply as biologics, which could be proteins, acellular matrix, even unviable things, or bone, for example. So is it biologics or biological material? And then some definitions imply it preservation at the liquid nitrogen temperature, because that has been the common cryogenic refrigerant, but that doesn't mean preserving in LN2 is the only way to cryopreserve. And some definitions might refer just below minus 80 Celsius, coming from the background of slow freezing history, which has been in the cryobiology field. So, and I took some of these examples, definitions from the papers published in past. You can see the top one. Cryopreservation is the process by which cells are maintained in low temperature at inactive state so they can be revived later. And that low temperature is said minus 70 to minus 80 Celsius. But that is not the case for a lot of vitrifiable organ CPA concentrations. For the definition at the bottom, for example, bottommost is cryopreservation may be defined below minus 80 and typically below minus 140. And here they're talking about all biologics. So even the experts within the field, that could be misleading, especially if someone is coming from outside. And the improper use on a general audience to a public would be if they're using cryopreservation as cryofreezing or cryotherapy or cryosurgery, cryogenics, cryonics, medical hibernation, or cryolipolysis. You name it, there is a term. Vitrification, now you're talking a little more deeper. And for example, the sources of confusion could be, it might sound like freezing, vitrification, glass formation, but it is ice-free. And assumption of that complete ice-free state may not be true in practice too. You may have microscopic ice, which you can't even detect using even your advanced imaging as such as CT. And or for example, a really common confusion could be non-equilibrium vitrification and slow freezing, because in slow freezing, you can still have glass and ice is permissible in the extracellular space. So you have a mix of glass and ice. Does it vitrify? Is it still vitrification or is it not? And then definitions can vary among the fields. For example, if you go into plant or horticulture and type vitrification, they will be using vitrification as a way to inspect morphological disorders in their plant tissue stems and so on. And that's where this definition on the bottom right, if you read, or actually the top one, is where the first definition is describing organ and tissues having a normal morphological appearance and physiological function, whereas the second definition is talking about the physics of it, which is transition from liquid to a solid-like state, with sufficient high viscosity. So different fields can define a same technique as different, not same, and which can lead to confusing terminologies. Example of improper use for vitrification might be freezing, which is not, supercooling, or solidification. For example, you might see glass is solid, but it is not a solid. It's a solid-like state, but it's still a liquid. And crystallization, if you might say, is glass crystallization. Now, is glass actually a crystal? No, it is not. So you are really seeing things which are confusing. So that is just two examples, but you can go to the research paper, which we just published in the CPP group, and we took a lot of examples of definitions within the cryobiology, which has divergence in the literature. And these conflicting use is gonna be creating barriers when you're starting to create a standard or to translate this technology. And for example, FDA needs to use one of these technologies, but there is not a clear definition for it or a clear consensus, which can confuse things, or talking between different groups. So for example, an conservation biologist talking to an engineer, and someone talking about, for example, diffusion versus equilibration. Both fields may mean differently, and that could lead to, again, confusion and sort of failures, unwanted failures. Lack of consensus also will hinder governance and regulations, as I said. And then commercialization definitely needs in standard protocols, standard terms. Even samples should have standard parameters, which needs to be listed on a cryopreservation protocol. So there is out there existing cryopreservation standard? There are some, but most of them are developed for either cell therapies or I should say, more into the slow freezing protocol, which has been widely adopted in biobanking. So, for example, ISO has two official documents out there for biobanking for mammalian cell lines. NSI and USB has chapters on cryopreservation of cells, but none of them covers what ATP-Bio is bringing to the table, which is these rapid vitrification technologies, rapid rewarming technologies for cellular 3D cells, organoids, spheroids, as well as for tissue slices and whole organs. So there is a huge unmet need for these terminology standards, which can cover tissue, organ, and whole organisms. And that will keep on growing as we are finding new breakthroughs in the field. So one of the ways, how do we start creating a consensus, is we look, how does other field have done it? And for example, ISO actually have a standard to create terminology work, principles, and methods. So that could be, sum of part could be adopted to create a working model, or Delphi method and ASME has their own process for creating standards. But if you look all three, it boils down to this flowchart, which we put, that you have to create and identify a list of terms so you can focus, and then you have to invite stakeholders. For our case, cryopreservation, especially the technologies coming out of ATP-Bio, these could be a list of stakeholders, general public, starting from whether it's a cryopreserved, let's say cryopreserved shrimp or a cryopreserved kidney. The general public is the user, or hospitals, OPAs, cryobiobanks, as well as botanical gardens, the people who are doing the protocols, whether it's engineers, life scientists, then regulatory bodies, FDA, USDA, and so on. And of course, the professional societies, whether it's Society for Cryobiology, Society for Low Temperature Biology, ISBR, and etc. So invite these stakeholders, the experts from stakeholders, create working groups from each of these experts. And then after you have created a consensus among these working groups, you seek an external review outside your group, reach a consensus, and that could be using a Delphi survey-based model, for example, to a survey or a special session at one of these cryobiology conferences, and then you publish a standard which would be a living document, and that's why you have that continuous updating and improvement as you find new and new breakthroughs in the field. So that is one model, but definitely we have started to look and create this sort of interest among the two different societies I talked, and we presented at both of them last year. This is the paper which I was talking about. If you're interested to more learn about, is this a really problem which will be in the field? I hope this paper convinces you this might be a worth looking problem. And so that's what the webinar is, basically a call to action to agree within our ATP-Bio and all of the cryobiologists working within the ATP-Bio, as well as starting to go beyond it to so we can speak the same language. And with that, I would say this is not just your webinar, but it has to be like an open panel model. That's why please give your thoughts, but to acknowledge all the people I'm working with for this, it's the ethics and public policy team, a lot of clinician engineers and ethicists, lawyers, and funding sources, NSF ATP-Bio, NSF Net Ethics, and yeah, Susan and Korkut who had been the leader for this effort. Thank you.
Yep. Thank you very much, Dr. Gangwar. That was very sort of provocative. We'd now like to have the PI of ATP-Bio give a few of his thoughts about this. Dr. John Bischoff, would you like to say a few things?
Thanks, Tim, and thank you, Lakshya, for that very nice presentation and also leading that nice paper that just showed up in Cryobiology. Yeah, as I was listening to you go through your talk, and of course I was thinking about this during the paper as well, you know, the need for standardization, you know, why is this important, or why is it important now? And actually, I'm going to reflect on a meeting that actually Lakshya and I just had today with a different group. And I think you'll see immediately why this is so important. This group has about $100 million that they have secured. It's called the Talion Institute, and they're going to be studying how to actually how aging occurs in mammalian systems. So they're going to be looking at a variety of different mammalian systems across the world, many different species, many different tissue types, 20 different tissue types from every system. And so we were discussing with them, you know, biobanks and standards and protocols, many of the same things that Lakshya brought up in his talk. And it became really clear that there isn't a clear understanding of standards or protocols. We started sharing what we understood, you know, in terms of opportunities for them. We started talking about some of the work that Matt Powell-Palm is doing, which is really exciting in the field, CPA discovery and coming up with new CPAs. They're thinking about, you know, well, what about CPAs that already exist in the field? And, you know, there's no standardization around that. There's no standardization around CPA discovery yet, or what CPAs to use on what tissues or what cells. And that got me thinking back to, you know, some of my really early days in the field where, and unfortunately this still comes up, where people are like, well, cryobiology is, there's no science there. It's 10% DMSO and 1 C per minute, and that's it, right? And unfortunately, I think that's true, that people don't know that there's good science going on and that, in fact, different cells and different tissues and different systems need to be treated differently to get good outcomes. And a great example of that is, I don't know whether it was maybe 15 years ago or so, maybe 20, the NIH and a variety of other federally funded biobanks discovered that many of their, and Susan probably knows this well, is that, many of those specimens that they had locked away using quote unquote standardized approaches, probably 10% DMSO and 1 seed per minute, were horrible. They have to throw them out. They were unusable. So, I mean, this clearly is a call to arms, as Lakshya was saying, is that we really do need to think about how we are going to, as a group and as a field, come to a point where we do have standards and protocols and ideas that can help people who are we are the experts, right? But like, how do we guide people who are not expert that are coming into the field or want to use the field to do the next thing, whether it's cryopreserving an organ or having tissue slices that can be available for doing multi-omics discovery of how we age or the next drug discovery. So I just think that this is a very timely paper. It's a very timely discussion. And if we're not going to lead this discussion, I'm not sure who will. So with that, maybe I will pass it back to you, Tim, and I look forward to a robust conversation also with the rest of the audience, who I know has questions, including Glenda. I saw yours in the chat. So looking forward to talking about that.
Yeah.
Thank you.
Thanks, John. Appreciate your thoughts. And I think the need for some structure is going to be clearly evident. Now we'd like to welcome my co-director for the EPP, Susan Wolf, for her thoughts on this issue as well. Susan?
Thanks, Tim. I agree with Lakshya and with John that this is really a pressing problem in this field. Let me put it in context for just a second. I think there are many domains of emerging science and technology where this problem erupts. There are lots of labs doing their own thing. Sometimes that's why there's a lot of terminological confusion. But part of the field maturing is coming to some agreement on terminology so that the science and its application can pick up speed. You advance reproducibility and replicability, as Lakshya argued. Labs can talk to each other. They can exchange protocols. Their data speak to each other. Their papers speak more directly to each other. You lay the groundwork for some kind of regulatory progress and ultimately successful deployment and commercialization. But I think there's a especially urgent problem in this field. And that is because we are talking in the advanced biopreservation world about building supply chains involving lots of actors over space and time, sometimes a great deal of time. John's example of the biorepository where he had to throw a lot of things out is an example of failure over time. But the more actors you have involved, who have to count on the prior actors, know what they're doing, pass the ball, successfully biopreserve material, have it stored properly, and then reanimated, again probably transported and used, the more you absolutely have to be speaking the same language. You have to know what you're dealing with. Any failure of communication along that chain endangers the viability of the material. And it's particularly urgent, I think, here because of the medical applications, where people's lives actually are on the line. The research, but also, everybody on this call knows that biopreservation of organs for transplantation has been a major focus, not the only, but a major focus of ATP bio work in progress, as Lakshya said. There's no way that clinicians and patients can possibly trust the material that we're asking them to trust and have placed in their body without this kind of confidence that everybody's speaking the same language along that full supply chain. There is no gap, and the standards are in place. So I did see Glenda Roberts' question about who's the right body to set the standards. And I think that would be I'm going to nominate that, Tim. Glenda may want to elaborate as a great question. In the paper, as Lakshya suggested, we called for the Society for Cryobiology to play a major role, a major convening role. But this is a great group to talk about, is that the right answer or not the right answer? Whoever it is has to be able to convene, command, build consensus, and succeed in what could be a very complicated process. So with that, I'll turn it back to you, Tim.
Thanks very much. Glenda, do you want to say something or not about that in terms of the exploration of where I have a question too, by the way, the exploration of who should be the standardizing group?
No, I think Susan summarized my question quite clearly. And after I looked at the presentation, it became pretty clear to me that we probably haven't decided as a industry who is appropriate.
Yeah, thank you. So this is going to be for both John and Lakshya. Having gone around this road a little bit, I'm very curious about the amount of disclosure that needs to be in our language. There's an awful lot of intellectual property and protection of ideas that go inside of this field. And the definitions of what we have, particularly for the agents which are necessary to prevent ice crystallization, oftentimes have lots of shortened names, and they're not always disclosed as to what's actually in them. And so for the cryoprotective agents which are out there, how much do you think that we need to concern ourselves about protection of intellectual property versus the disclosure of what the actual composition of our CPAs actually is?
Who has to go on? Do you want to jump, Lakshya, or you want me to go first? What do you want?
How about you go first and
Okay. Early in my career, I was just learning, like we all were, like how to use the web, right? So one of my early web searches was cryobiology patents. And what was really interesting was the vast majority of those patents were cryoprotectant cocktails.
Right.
And to your point, Tim. And so, and now we're in this moment of CPA discovery again, which I think is super exciting, by the way. Matt and Adam Higgins and Becca and others are really, leading the charge there. AI is being part of it. And I think it's very, very exciting. But we're also in this moment where all of us, especially I can still remember when I was interviewing for my job here at the University of Minnesota, and the guy who was the head of the search committee, he was like, well, are you more of an academic or are you more of a businessman type? Basically that was the question. And I was like, well, I'm an academic, I didn't come here to run a business. And he said, well, John, more and more of us are running our labs like businesses, and that, you're going to have to deal with that. And this guy went on to form Fluent, which is like one of the most famous finite element software packages in the world. And that's the truth. and Matt, I think, is dealing with that. I think all of our young faculty are dealing with that. I'm dealing with it, which is that, we are, we're not being forced to create IP, but the incentive structure is there for us to create IP. And, so it creates this tension that you're talking about, Tim, which is that we want to publish. And that's also a huge driver, right? But we also want to then have IP. But then there's the disclosure question that you asked. And I think ultimately you're going to be forced to disclose. If you're a company that licenses that technology and you want to put it through the FDA, you will be disclosing it. But I think intrinsically in your question, you're asking, should we be disclosing beyond that? Isn't that your question, really?
Yeah, if we are going to standardize language, the question is, what is it that our language is to talk about? One of the very major components of cryobiology is the fact that we can sometimes need to introduce materials to prevent ice nucleation and damage from crystallized material. And so how much do we need to disclose what we put in there in order to do our function? Now, there's all sorts of things in terms of thermodynamic changes and heat transfer, and there's a zillion other things which go on. But the one, just like you noted, that keeps getting my attention, which doesn't get disclosed inside of the papers that we write, are the composition of the cryoprotective agents that we use and their combination. And that is an intellectual property protective component of what we move forward on. So yeah, that is sort of what the question was.
Yes.
And can I ask a question on top of that? Is there an animating concern behind this question that building harmonized terminology is a cooperative effort, but intrinsically, labs are actually in competition with one another? Is that a barrier?
Why would that be a barrier, Susan?
Well, I would hope it would not, Mehmet. But, I mean, as you know, Terry Tiersch, I don't think Terry's with us. If he is, he could speak up. He's one of our ethics and public policy panel members, wrote a wonderful paper in his field of aquaculture with Torres about building a collaborative scientific community in his field, using all sorts of really interesting things like open source, 3D printing, tons of sharing, even beyond terminology. And so something like that, going that far beyond shared terminology, I think does create a tension with the competitive streak.
What I've seen, Susan, way back, for example, in biomechanics, when the field was just growing, and there were different, what I call clubs. Some people believed in certain terminology versus another. And there was, some arguments back and forth. And I could see things like that, but I don't know if there'd be any competitive reason. It's more like scientific. For example, what is the definition of vitrification? How do we try to be very disciplined. Usually we say apparent vitrification the first time is because it's really, unless you have serious measurement with DSC or XA and whatnot, you don't know if it's vitrified or it's partially frozen. And so the terminology is very loosely used, especially those who are so I've seen those kind of arguments back and forth in other fields, but I don't think it will be because of competitive reasons. I might be wrong. I hope I am not.
Well, I would just say, being a friend of Terry's and having had, sort of many interesting discussions with Terry, there's not a lot of, how shall I put this? That, it's not, so if you take healthcare, organs, organoids, and things of that nature, There's a lot of money at play. There's probably less money at play in aquaculture. And so I think there's a little bit more of a willingness and maybe even a need for sharing in Terry's community than I think there is in our community. And I think Terry would probably agree with that. But one of the things that I think is really nice about our community here in ATP-Bio that Mehmet keeps bringing up is that we seem to be able to live within that competitive structure, especially the healthcare structure, with multiple companies that have been formed. And the fact that Matt is going after one CPA, we're going after a different CPA, and we're like friendly colleagues, and we talk about, doing collaborative work together. And I think, at least for now, we've managed it is not a hindrance. I would say it has not been a hindrance to what we're trying to do collectively, which is nice. And I've certainly seen it in other spaces where it can be like quite damaging to the field.
Do you think there's a reason to call for some sort of minimal standard of disclosure regarding at least the CPA field?
I mean, I think there already is some push for that, right? I mean, but I will bring it up, and this is maybe a conversation item, right? DMSO, dimethyl sulfoxide. Some people will call it, toxic, that there's neural toxicity, there's this, that, and the other. And so people have been looking for maybe a decade or more for like DMSO substitutes. And I mean, I think that's fine. And, it's certainly, it's a subfield now. And some of us in the field are have formed companies around that. And so I think that there's a need to sort of declare that. But then on the other side of that, if you're in the organ space, I don't know anyone who's gotten away from DMSO in an organ system in terms of being able to actually vitrify a bulk system. And I think I saw Greg on the call. So I don't know if he's still on the call, but Greg Fahey, Greg uses, we all use DMSO. It's a very effective glass former. And there he is. Hey, Greg. I don't know if you want to comment on that.
Well, yeah. So I think everything depends on context, right? I agree with you. There is no organ vitrification solution that seems to be very successful that doesn't use DMSO right now. But we're not freezing single cells like other people are doing. So I think it all depends on what your application is. So I think probably all that I have to say on that.
Okay, everyone, Greg agrees with me.
By the way, if you guys haven't seen, Elias put a link to an article on terminology, nucleation, and ice formation. It is really very nice. Like quick, I wasn't aware of it, but it's very relevant.
Yeah, that's great. Thank you.
Yeah, and Tim, like if I can put my thoughts back to your original question, like what is all we need to declare or provide in a cryopreservation protocol. So that would depend upon the intended use case. And one example which I can see directly is if you want to reproduce a protocol, then you will need to give all the specifics of how you're cooling, what is your container geometry, how much volume you have to put in, what temperature you will cool to, what the CPA composition is. And so then only you can reproduce in that way. But then how do you sell or how do you protect your IP, right? So I was thinking about that as how ASME, American Society of Mechanical Engineers, they do a lot for boiler codes. And that is safety codes because these are high pressurized boilers. And a lot of even in nuclear power plants as well. So, but to provide the guidelines, not only they give all the steps to create a high pressure boiler, but when they're leaving some of, let's say, someone's protocol or someone's manufacturing design, they provide it in a form like 5X. So it's an inherent safety factor provided to prevent any disastrous failure. So that 5x sort of safety factor could be when you're providing a CP cocktail and you don't want to, like, disclose all, then in terms of safety considerations, you not only show the data on your CP cocktail viability, but you could show like what is the toxic level of these, which you should not exceed. And then, like, you're providing at least some sort of safety net for people to not go beyond, even if they don't know what is in that cocktail or what is the exact mixture and so on. So yeah, that was kind of one thought I was thinking, where you could start defining sort of border of safety nets around your cooling geometry, your cell temperature, exposure temperatures and all, so that you can prevent some catastrophic failure.
Yeah. I mean, we obviously need to protect from catastrophic failure in this, and the importance of some standardization somewhere is important, but it's the tension and the balance. Are there other comments? I don't.
Well, I wanted to raise something, Tim, which is, I think it's really important to think about the range of perspectives that need to be represented at the table for this effort to come up with consensus terminology, because there's such a huge range of stakeholders and potential users. And if you think, for example, back to organ transplantation, the terminology, I suspect, could have a very big impact on adoption of innovation by clinicians and acceptance by patients. I mean, if you said to me as a layperson, okay, well, we have this traditional allograft, this fresh organ, but we've also got this frozen organ, evoking the frozen section of the supermarket. Which one? The terminology is going to bias me in terms of my acceptance. I'm going to go for the fresh. So I think that's one example, but it actually could be a very important example of making sure we have all the folks we need in this conversation. So I think if it becomes a conversation convened by the Society for Cryobiology or the Society for Low Temperature Biology or one of the professional societies, one of the challenges will be inclusion and making sure that that obviously the terminology has to work scientifically, of course, but it has to work more than scientifically.
Yeah, but Susan, your example, which is a great example, by the way, I've been thinking a lot about that too, is like who gets the first frozen organ or cryopreserved or vitrified organ or whatever. There's a flip side to that, which would be like, okay, there are people that are currently not even, I think they can't really get on the list, right? Because they're too old or they're too whatever, or they might have heart and kidney failure or whatever, right? So what about those people having the opportunity potentially to take that frozen vitrified organ,
Well, let's go further, John. Maybe with the frozen organ, you've had a chance to precondition me and optimize my readiness for the organ. Maybe you've even done some advanced testing and manipulation. I can make those arguments too.
Okay.
And it may be a superior choice, but I still think words matter.
Sure.
Which is why it's important to have everybody at the table.
Sure. No, I was not arguing against that. I was just looking at your example and flipping it a little.
The other thing I'd like to ask, and I'm going to be a little provocative here. In this group of experts, Is the prefix cryo being overused?
Yeah, because the society is called cryobiology, but it is not just cryo, it has all kinds of other ways of putting living things into suspended animation.
Yeah.
But at the time it was intended for cryo and it expanded, and society always struggles with that labeling.
Well, I was going to say that I was going to respond to that as well, because vitrification goes beyond what we've been talking about. Vitrification also goes to what Mehmet was doing. He submitted a huge grant to this BOSS ARPA-H thing, which is all about desiccation. So there's a whole other field that's dealing with like essentially room temperature desiccation.
Or hypothermia. It's not cryo.
Yeah.
Well, I was really inspired, Lakshya, because you really advocated, and I think you were very right, to make sure that a section of this paper talked about all the ways, and not even all, but a big range of ways in which the word cryo is being deployed, including in the general public. And it kind of makes wrestling the terminology to the ground even more difficult. I don't know whether you have thoughts about this, Lakshya.
Yeah, and I think when I started, that was the thought. Why, like, this whole paper origin to me was like, everyone uses cryo very loosely, and it's just the most popular adjective. Even if it's an hypothermic machine fusion, you might like still send it to cryobiology and call it a biopreservation as cryopreservation. And the sort of bad side of it could be is like if you start having cryo as tied to in like things which don't work or which are like too much manipulated, whatever biological system you're saying, then people might start making a fear of like how GMO is, right? Like you go to a grocery store, buy organic, non-GMO label, and you take it as opposed to like a product which is GMO. So it could be like, oh, I don't want any cryo product in my body. I only want non-cryo, organic, fresh product in my body. And that could be because like cryo is being used so loosely and things which don't even work or don't show reversible preservation. Yeah, it just becomes like too vague at that point.
Does it make sense to create a living document like Wikipedia, where we put the initial set of common words and whatnot that most people agree, and then let the society actually contribute to that? I remember some years ago when Wikipedia first came out, people said, oh, it's going to be all kinds of errors. And then they looked at it against the Encyclopedia Britannica, and the error rate was lower because too many eyeballs looking at it and correcting things.
That's a very interesting idea. After I saw Glenda's post in the chat, I immediately went and Googled, and of course, it went to Wikipedia.
Yeah, here we go.
To your point. And and it talked about the Society for Cryobiology, SLTB, and basically there isn't one governing body, ISBER, et cetera, but it was very informational. And so, I've actually been, I think that the cryobiology Wikipedia page could do with a little refresh, and it could begin to address some of these issues to the benefit of all, actually.
Yeah, and so I think, I remember in I think 2020 when ATP-Bio got the fund, I went and edited in the ERCs to add the ATP-Bio on the Wikipedia page, because that's how I got my information from Wikipedia before the Google AI overview and whatnot. But there are professional societies like American Chemical Society, ACS, IAR, International Institute of Refrigeration. And many of them has a dictionary option, which you can like search. And I'll put in the chat, for example, IAR. So you can search any of the terms in their field, and it shows a definition, which I'm assuming the society has agreed upon. And so that's like becomes at least a more consensus way of knowing things. So a Wikipedia page, or if the society is willing to like, have one page in it, I think that would be a start. And I know when I presented back at Cryo in Hanover last year, a lot of people, and Elias was one among them, showed interest to actually work on this, and even wanted a session to organize and even put a survey on Cryo, like, Society for Cryobiology. You can have an ongoing survey for a year. Which are the 5 definitions seems most relevant or most incorrect to you? And yeah, but Wikipedia seems like easy, you can edit, but also then like, anyone can edit too, versus Society for Cryobiology or ATP-Bio's webpage, not anyone can edit and log in.
So here's a question I've been dying to ask John and Mehmet. When you were giving birth to ATP-Bio, when you were conceptualizing this thing, did you deliberately choose advanced biopreservation, not cryo? What was going on?
Yeah, go ahead, Mehmet. That was you. I mean, we agreed, but it was your idea.
Yeah, I mean, because it wasn't Cryo, wasn't general enough, and we also struggled because the ATP-Bio is really focused on living systems biopreservation, and not drugs or vaccines in general. Not that we are not interested, but it was more making living products accessible and managing their supply chain. So we tried to make it as general as possible.
I also really wanted it to work with the moniker ATP-Bio, so I just kept working with that really hard until it all worked, So that's my excuse. Cool.
And so there are, go ahead, Susan.
No, I think it was Glenda.
Just a quick question. Are there organizations outside the U.S. who are also focused on this issue? Or are they a part of our group here? Because I don't know all the people.
I just answered that question. The most prominent one is Society for Low Temperature Biology in UK, and many members are colleagues, friends, and they're part of ATP-Bio.
And the Society for Cryobiology actually is an international society. Yes. So there are like different arms in different countries. So they have the Japanese Society, the Chinese Society, Spain, and so on. So it is a very highly representative body. Yeah.
Right.
But we need help. So when we start talking about organs, we're going to have to come to Tim and FDA and people like that.
So I want to say thank you. We're almost at the very end here, and Ellie has flipped back up. So we're going to assume that we're coming to the time when we have to close this. Want to thank Lakshya for his superb discussion and for all the participants which came along. So Ellie, it's back to you.
Thank you, Tim. Well, as Tim said, this concludes today's webinar, and I'd like to thank everyone for joining. A quick reminder that we did record today's webinar, and past webinars are also available on our YouTube channel. We are excited to announce that the ATP-Bio public webinar series will continue through starting this fall and through spring 2027. And as always, the webinars are free and open to the public. If you'd like to view the schedule for year 7, visit our website at atp-bio.org. So that's it for us today. Enjoy the rest of your day. Thank you.